Hello biology students, and welcome to video 5 in the DNA unit. So remember from previous videos in this unit that protein synthesis, or the process of making a protein from a DNA message, happens in 2 steps. So in today's video, we're going to look at the first step, which is called transcription. And this is when DNA, the gene, is converted to an RNA intermediate message. And in this case, it's going to be converted into an mRNA. Remember, that stood for messenger RNA. And we'll look at the next step, translation, in a later video. Okay, so transcription. So here is the whole protein synthesis process. So you can see this is supposed to be the nucleus, and this is the cell, and this is the cytoplasm, this area here. So you can see in the nucleus, I have my double-stranded DNA molecule. And you can see it's been opened up right here, and one section is used to make a copy of RNA. So that's transcription. That's what we're going to look at in this video. That RNA then leaves the nucleus via a nuclear pore. It's found by a ribosome in the cytoplasm, and the ribosome takes the RNA and reads it and makes a protein. So that will be translation. We'll look at that in a later video. Okay. So transcription, what we're going to look at today. So this is when a copy of a portion of DNA is made into RNA. We're going to look at a simplified version where we're only going to look at one enzyme doing most of the work here, and that enzyme is going to be RNA polymerase. Okay. So transcription, it has 3 steps. It has initiation, and like it sounds like, this is where transcription is initiated, where it started. So this is basically how the RNA polymerase, that enzyme, is going to find where to start and kind of get going. Elongation is where I elongate or build my new growing RNA molecule. So you can see that here. I have an elongated or growing RNA. And then termination is where I'm going to stop transcribing. Okay, so the first step, initiation. So one thing I want you to think about, when I make an RNA, I'm only going to make a copy of whatever one gene I need to make, right? Because, you can kind of assume the cell at the time, it only needs to make one protein, so therefore it only needs to copy one gene. So I'm not going to copy the entire DNA molecule, because the entire DNA molecule is thousands of genes. So the first thing that has to happen to initiate or start transcription is the enzyme that's going to do the transcription has to figure out where it should start. Should it start on the end of the DNA molecule, somewhere in the middle, etc., etc.? It has to find the beginning of the gene. So the section of DNA that signals the beginning of the gene is called the promoter. So this is a stretch of DNA that acts as the start transcribing signal. This is where the transcribing is going to start. Okay, so what's going to happen is our enzyme, RNA polymerase, is going to bind to the promoter, which is a section on the DNA, unwind the DNA so it could read it, and then start transcribing. So you can see here's my DNA that's unwound, and maybe this section of DNA right here, that's the promoter, because that's where the RNA polymerase, the enzyme, first bound and gets ready to start adding RNA nucleotides. So the next step is elongation. So at this point, my enzyme is bound to the DNA, and it's ready to start reading it and build the RNA. So what it does is it reads the DNA message, and then it adds the complementary RNA nucleotide. That's why it's called RNA polymerase, because it's going to build RNA. Okay. So maybe if my DNA, maybe the DNA was TACGA. Okay. So what it's going to do is it's going to read this message and add the corresponding RNA base. So here's my DNA here. So as it builds the RNA, the RNA polymerase, where there's a T, it's going to add an A. Where there's an A, it's going to add a U, because remember, RNA has U's, not T's. Where there's a C, it's going to add a G. Where there's a G, it's going to add a C. Where there's an A, it's going to add a U. So it's going to use uracil instead of thymine, and it's going to link these nucleotides together. So here's now my new RNA strip. This will peel away from the DNA, so that way it can leave the nucleus and go find the ribosome to be made into a protein. And then when we're done, the DNA starts to wind back up. So you can see here, I'm adding my new RNA, and then once it's done, it detaches as the rest of it still is being built. And then finally, the last step is termination. So eventually we reach the end of the gene. So the DNA sequence, the part on the DNA that signals the end of the gene, is called the terminator. So maybe this section of the DNA right here is a terminator. And once the RNA polymerase reaches that section of DNA, it detaches, the RNA detaches, and the DNA winds back up. So you can see now I have my completed RNA, my copy of the gene, my DNA that's wound back up, and my enzyme that's ready to do another transcription somewhere else. Okay, so one thing you might have thought about or noticed already is that in transcription, we're only copying one strand of the DNA, right? So kind of the question is, what would happen if you copied both? So say here's my DNA. So say I unwind it, and let's pretend the RNA polymerase decides to make an RNA copy of the top strand, the one right here. Well, that RNA would be U, 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 A, G, 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 G. Okay? Now we can pretend that the RNA polymerase decided to copy this bottom strand instead, which is down here. And if it did that, the RNA would be A, U, 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 C, 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 C. So you can see from one DNA piece, I can actually make two different RNAs. So that's kind of cool. I mean, in theory, you could make two different RNAs and two different proteins from one piece of DNA, depending on which gene or what side you copied. So I just wanted to point that out. It's kind of a little fun fact. Okay, here's another picture of the same process. So you can see here I have initiation. This green part of the DNA, that's the promoter. The red part's the terminator. So the RNA polymerase during initiation is going to bind to the promoter. During elongation, it's going to zip along, and it's going to make the complementary RNA. And then when it gets to the terminator, it's going to fall off, and the RNA will be complete. And just as a reminder, it builds this RNA using the complementary base pair rule. So where there's a G in the DNA, it's going to add a C. Where there's a T, it's going to add an A. Where there's an A, it's going to add a U, etc., etc. So it turns out we're not done here, because this form of the RNA is just like a rough draft kind of form. It turns out that this form of the RNA is not directly used to make protein. It has to be edited and processed first. So the first thing that happens when it's being processed is I add a cap to the 5' end of the RNA, okay? And this cap is actually just a single G nucleotide. And I also add what we call a tail to the 3' end. So basically we add between 50 and 250 adenines, A's, to the 3' end of the RNA. And the reason we do this, or the reason the cell does this, these parts are not actually made into a protein, but they help the RNA leave the nucleus properly, they make it more stable, and they help the ribosome to bind to it. So they're kind of like, similar to those chemical addresses that the Golgi added to the protein during protein production. They just, they make it more stable, and they help it get to where it needs to go. Okay, and then finally, the last part of RNA editing and processing that we do is this weird thing called splicing. And basically, the idea is that a piece of RNA is not all of it gets used to make a protein. So maybe here's my RNA, okay, that I just made via transcription. And here on the 5' end is my little cap. And at the end, I have my little tail that I just added. And it turns out that maybe this part is used for processing, for making a protein, but this part here is not. And maybe this part is used, and this part is not. And this part is used, okay? So the parts that are used are called exons, and the parts that are not used are called introns. And these names make sense because exons stand for expressed regions. These are the regions that are going to be expressed into protein. And introns are the in-between non-use sequences. So what basically happens is for the mRNA is going to get cut up, the introns are going to get removed, and the exons are going to be spliced, which means joined together. So here's a picture of that. So here's my original DNA. So here's transcription, and then the cap and tail being added. So I copy this DNA into RNA. I add my 5' G cap. and I add my 3' A tail, and then I cut out these introns, these pieces that aren't going to be used, and then I splice together the exons. Now I have a shorter RNA, and this is the mRNA that's going to be used to make the protein. So this mRNA leaves the nucleus to go to find a ribosome for translation, which is going to be our next step in the next video. Okay, thanks for watching, guys, and I'll see you in video 6.